Tēnā koutou katoa, welcome back. This is the last section of the lecture for human cells. So I'm just going to recap briefly what we've been through with the organelles. There was a lot to take in there, hopefully from having drawn the picture and engaged in that, and then going over it again here. It'll help it kind of sink in a little bit. There's a lot to remember. I don't expect you to remember everything after having watched these lectures. The nice thing is that you'll have these to come back to. Feel free to watch them as many times as you want. There's a few other really cool videos that I'll put up here as well that may really help you kind of conceptualize and visualize these things that we're talking about. Okay, so we're just going to recap really quickly and then move on to having a look at active and passive transport and what the difference is. So we'll just put it in table form. So again, these are just the key points from the slides that we've gone through up until now. So remembering cell membrane contains the cellular contents and regulates what comes in and out. Okay, so it's like the bouncer at the door going, oi, you, no, yes, you can go. You can go. And basically just it keeps the structure of the cell. Cytosol surrounds and supports the organelles, medium through which nutrients and waste move. So remember, that's just a gel-like substance that allows things. It's where the organelles are, where the organelles sit within the cell, and it's our medium for which things move through and around in the cell. Nucleus contains genetic information, control center, brains of the operation, basically controls all of the cellular functions. Okay. Endoplasmic reticulum, so it's a transport mechanism. Rough contains ribosomes where proteins are synthesized, and smooth, so that's without ribosomes, and that's the site of lipid and steroid synthesis. Struggling with that word today. Okay, so basically rough endoplasmic reticulum, that's where proteins are made. Smooth endoplasmic reticulum, that's where lipids and steroids are made, and the endoplasmic reticulum itself, it's a transport mechanism that helps move things from there out into the cell. You'll remember that the Golgi apparatus helps in protein synthesis. So packages protein, puts its finishing touches on it, packages it up, and then sends it off to where it needs to go. So remember, that could be within the cell or it could be outside of the cell. But that's the job of the Golgi apparatus. Mitochondria produce adenosine triphosphate, ATP. ATP is easier to remember. Gives energy for cellular function. So it gives energy for all cellular functions right throughout the human body. It is the only currency that the human body knows how to use. Okay, so we have multiple mechanisms to convert those stored nutrients, so carbohydrates, proteins, and fats. We can use all of those three things, and alcohol, to produce ATP within the body. Okay. Last slide here with this one. So ribosomes, remember, they are the sites of protein synthesis. They can be free, so floating around the cell, or they can be fixed and that's attached to the endoplasmic reticulum, so the rough endoplasmic reticulum. Lysosomes, okay, those that might be a little bit confusing. So remember, lysosomes are the housekeeping. Phagocytosis is just a fancy word for bodies getting engulfed within that lysosome and then taken off to be excreted out of the cell. Okay, so the enzymes, remember, they had enzymes inside them that help break down foreign matter or whatever it happens to be that they've engulfed. Okay, so phagocytosis is just the process of a lysosome coming along and Pac-Manning, grabbing onto the foreign stuff that we don't want and then transporting it away out of the cell. Centrioles, remember, so their job was to separate chromosomes during mitosis. Mitosis, that's just cell division. Okay. And then we've got the three projections on the outside, which we talked about. We had those little animations. So cilia create movement over the surface of the cell, so they physically move things across the cell membrane. Flagella they actually physically allow the cell to move. So the examples were sperm and bacteria, and microvilli increase the surface area to facilitate absorption. So particularly important in places like the digestive system, where we want to be absorbing as many of those nutrients from our food as we can. So microvilli within those cells of the linings of the digestive system really help us in maximising that. Alright, so that was just a quick recap on the organelles. What we're going to do now is move on briefly to transport. So active and passive, we're going to talk about two different types of transport. Essentially, this visual was a really good one, I felt, to help people understand what we're talking about. So passive, downhill, in the sense of cellular transport across a cell membrane. So it will happen without any input, and it will happen from substances will move from high concentrations to low. So for example, if there is a huge amount of sodium within a cell and not very much outside it, the natural tendency then would be for that sodium to diffuse from inside the cell to outside until we kind of really reached equilibrium. Okay, so we'll talk about that in a little bit more detail. So active, uphill, normally moving from low concentrations to high. So this is where we physically need to add some energy. You'll see that little three-letter representation there, ATP. So remember, that's the energy we use, the energy currency within our body. ATP is used to get things to move against that natural gradient, so whether that's concentration or pressure or temperature or whatever it happens to be. So if we had the same cell that had lots of sodium inside and not very much outside, but because of the function of the cell we wanted to move that, continue to move more sodium into the cell, then it would be an active transport mechanism, and it would need energy, it would need ATP, because you're going against that natural kind of diffusion gradient. So, passive transport, it's called diffusion. I mentioned it in the last slide. Movement of a substance from an area of high concentration to low. So remember think about that as downhill, doesn't need energy. Okay? Kind of diagram A there shows us, if you put a tablet, like if you dissolve, say, a Disprin tablet or like a Barocca in a glass of water, start with the tablet and it's got all the substance in there. You drop it in, there's a reaction, it diffuses until eventually there's no tablet and there's just a substance that has got an even concentration of whatever was in the tablet. So whether it was aspirin, whether it was B vitamins or whatever. Okay, so that's an example of diffusion. The other example of diffusion there is a little bit stinkier. So again, high concentration to low. Equilibrium, that's the point where there's no longer any difference in concentration. So if we're using cells as an example and talking about transport across a cell membrane, if we had our cell that had sodium on the inside and not much sodium on the outside, and so the natural gradient was taking it and diffusing that sodium from inside to outside, there would be a point where the concentrations were equal, and so then no natural diffusion would occur because the concentration is the same on the inside or the outside. Right. The last one we're going to look at is active transport. So thinking back to the picture that we saw in the first slide there with the downhill and the uphill, so passive transport or diffusion is downhill, so it follows the gradient, the concentration gradient. So from high concentration to low will naturally diffuse across a cell membrane. Active transport, where we need pumps and we need ATP, is that uphill, so we're going from low concentration to high. So obviously the natural tendency is for the substances to go that way. We want them to go that way, we've got to give them energy. Okay, the example on this picture is potassium ions. which are represented by the little blue circles with K plus on them. In this instance, they're being pumped inside a cell, even though there's already much more inside the cell than outside. Okay, so that natural gradient in this particular picture would have potassium moving out of the cell, but if we want it to move on, we've physically got to pump it in there. Okay, an example of this in the human body would be in the nervous system, where there's sodium and potassium pumps on the nerve within the nerve cells that help replace those substances to resting after a nerve has passed an action potential. So you don't need to know the details, but it's an example within the body that uses active transport and these pumps. So things to remember: low concentration, too high, so against the natural concentration gradient, and they require energy. So, and again, the currency for our body is ATP. All right, so that's pretty much all the content that we need you to know for this cell section. From here, what I'm going to do is the next video will be a bit of an instruction or a how-to for the assessment. So the assessment here is a series of online quizzes, the first of which will open at 9 a.m. Monday morning, so and then run until 9 a.m. next Monday. So you'll have an entire week to do the online quiz that's related to the musculoskeletal system, so the content we did last week. It is not the quizzes on the Moodle page that are between the lectures. It is, you need to go to the assessment portfolio, move down to task 8, and you'll see there is 10 quizzes. They'll be open for the whole week. You can have as many attempts as you want at them. There's 2 parts to it, and I'll explain that in the next video. So I'm going to leave it there. Stay safe out there. If there's any questions with this content, don't be afraid to kind of jump online and contact us via Moodle message or via email. But there is, utilize the resources that you've got here. You can watch these videos as many times as you want. You can, there's some other really cool resources, Quizlets and such things to help you with the learning that goes along with this. And I guess the beauty of the online space is that you can do it at a time that really suits you. So depending on what's going on within your bubble, with your whānau, that might be first thing in the morning, it might be in the evening once the kids are all in bed. It might not matter to you if it's just you and your partner or you and your flatmates in the house. So, but you can come back to it as often as you need to. But there's some pretty cool resources and videos. So have a poke around with the stuff that's that accompanies this. And then once that assessment video is kind of done and dusted, there's Vicky's put together a couple of really neat videos and quizzes related to mitochondrial disease, mitochondrial dysfunction, and haemophilia that relate specifically to cells in terms of conditions. So again, that's the conditions that link to AMP here. Are some of the ones that you might get expected to do when you're looking at preparing your assessment task 2, so your creative composition. Right, more on that later. Ka kite anō. We'll see you all soon.